This is a video for Unit 1 Biology on the structure of nucleic acids. So our learning aims for this video are to describe the structure of nucleic acids, including DNA and RNA, describe how polynucleotides are made in condensation reactions, and describe how the double helix is formed through complementary base pairing. So first of all, what are nucleic acids? Well, these are biological molecules which include DNA, deoxyribonucleic acid, and RNA, which is ribonucleic acid. So DNA we're going to look at in more detail first. This is the genetic material and it contains all the information needed to make and control individual cells and whole organisms. Its main role is to code for the primary structure of proteins, and we're going to be talking about that in a little bit more detail in one of the future videos, but today we're going to be mainly focusing on the structure. So nucleic acids are polymers, and the monomer is known as a nucleotide. Nucleotides are all made up of 3 separate components. So you have a pentose sugar, a phosphate group, and a nitrogen containing base. So here's a diagram just to show you that a little more clearly. You have your phosphate group, your pentose sugar, which has 5 carbons, and your organic base, which is nitrogen containing and can be either adenine, cytosine, guanine, or thymine. So in DNA, the pentose sugar present is deoxyribose. You can also have a ribose sugar, which would be present in ribonucleic acid. And if you look very closely at the 2 different structures, you can see there's just a small difference I'm highlighting here. The deoxyribose has 1 less oxygen molecule there. Now if we look at the nitrogenous base in a bit more detail, so we have got 4 different nitrogenous bases. They are adenine and guanine, and if you look closely there you can see that both of them have a sort of like double ring structure. So they are known as purines, whereas cytosine and thymine, they have a single ring structure. These are called pyrimidines. Okay, so again, just to sort of confirm that, you've got 2 main groups, purines, which include adenine and guanine, or A and G. These both have a double ring structure, and the pyrimidines, which are thymine and cytosine, and they both have a single ring structure. So now we're going to look at how the nucleotides join together to form polymers, and this happens by a condensation reaction. So if you've studied other biological molecules, you might already be familiar with condensation reactions. In this type of reaction, water is produced. That's what makes it a condensation reaction. So here is a very, very simple diagram to show the 2 nucleotides. When they join together, a link is formed between the pentose sugar of one molecule and the phosphate group of another, and also water is produced. So that's quite a basic diagram. On the next slide, I'm going to go into a little bit more detailed explanation. So this is just the full drawn out chemical structure of the molecules. So again, don't be too put off here. It's just to show you that when you have a condensation reaction between 2 nucleotides, in this case adenine and guanine, you should be able to see the highlighted section here between the pentose sugar and the phosphate group of the guanine, and I've highlighted the 2 hydrogens and the oxygen, which are then removed from the molecule to form H2O, and you are left in the product with this link here between those two molecules. So you now have that bond, which is called a phosphodiester bond, and you also have the H2O, or the water, produced there. So that's just to show you in a bit more detail, and it's quite useful to know where the water molecule is being removed from in terms of the condensation reaction. So then what you end up with is a long chain of nucleotides, or a polynucleotide chain, and you can see the phosphates and the sugars sort of all the way along the edge there. This is often referred to as the sugar phosphate backbone. So they alternate as you go down. You have phosphate, then a sugar, phosphate, and then sugar. And now we're happy with that, we're going to have a look at what happens in terms of forming the double helix. So to form a double helix, this is where you get complementary base pairing. So hydrogen bonds form between a purine base, so either A or G, and a pyrimidine base, either T or C. They always pair up in the same way, and this is called complementary base pairing. So you always get A pairing up with T and C pairing up with a G, okay? And you can see again in the diagram that that hydrogen bond is formed between them to make the double helix structure. So here in this slide, we're just showing a few more examples where they're all joined together in a chain. So you can also see here that between adenine and thymine, you get two hydrogen bonds linking them together, and cytosine and guanine, they link up with three hydrogen bonds between them. So when this happens, you then end up with a double strand of DNA, which gives us that classic sort of double helix structure. So now we've talked a bit about DNA structure, we're going to move on to RNA. So RNA is also a polynucleotide. It's made up of lots of individual nucleotides, and again, each of them has three components. They all have a phosphate group, a nitrogenous base, and a pentose sugar. So that all sounds very similar to DNA, but there are a couple of key differences. So the first one is in the nitrogenous bases. So your cytosine, guanine, and adenine are the same as in DNA, but instead of thymine, you now have this additional base called uracil. So that replaces thymine in the RNA. So that is one key difference there, and we've also just highlighted that here for you, so that you know instead of having a T in RNA, you have a U. The next slight difference is the pentose sugar. This is the ribose, and again we looked at the structure of them earlier in the video. And the final difference is that RNA is usually single-stranded as opposed to double-stranded. So here in this diagram, you can see that where DNA has a double helix structure, RNA is normally existent as a single strand. So those are the key differences between DNA and RNA in terms of their structure. So that's all we need to know about the structure of